thank you. Today I'm going to be introducing you to a DNA design method called Ledidi. But before I do that, let's take a little bit of a step back. Over 20 years ago, the first draft of the human reference genome was released, and a little snippet of it looked like this. In the words of Dr. Eric Lander, genome, bought the book, hard to read. Even though it is challenging for us mere mortals to understand these long sequences of A's, C's, G's, and T's, having this reference enabled the next generation of biochemical assays to be developed. Here, we can see the readouts from 4 of them, indicating that there's at this enhancer here, there's the binding of OCT4, SOX2, and NANOG, but not necessarily the binding of KLF4. Clearly, these sorts of readouts are invaluable for understanding regulatory genomics. However, relying solely on these readouts is reminiscent of the parable of the blind men trying to describe an elephant based on what they felt. Just because we understand what is happening and where in the genome doesn't mean we understand why it is happening there. So, enter sequence-based machine learning methods. There are many different computational units that you can use internally, convolutions, transformers, Mamba, whatever. And there are many different ways of phrasing the exact task that you have the model solve. But broadly, these are models that take in nucleotide sequence and make predictions for these sorts of readouts. However, in my view, the predictions from these models are kind of the least interesting aspects of them. After all, most of these assays provide us with genome-wide readouts already. There is no more secret genome that we need to make predictions for. Rather, the strength of these models is being able to run them backwards to get them to explain why they made the predictions that they did. And so here we can see that the reason it's predicting the binding of OCT4 here is because there's an OCT-SOX heterodimeric motif right here. We can adapt these methods in order not to understand the genome like this, but to design regulatory DNA. So imagine the situation in which you have an initial template sequence, like before. You have your machine learning model, but now you have a desired output from the model, something that you would like the model to predict. You want to change it so that it does that. You take your sequence, you run it through the model, you get your actual output, you compare that to the desired output from the sequence, and then you run that backwards through the sequence. Rather than highlighting the nucleotides that are driving model activity, like we saw before, you now highlight the nucleotides that need to change in order to achieve your target objective. Now, here we encounter a little bit of a technical issue. Before, the attributions that we were looking at were continuous valued. They were varying heights, with the higher characters meaning that they corresponded more to the model predictions. But here we need to make a discrete choice. Which base pairs do we edit in order to achieve that objective? Ledidi rephrases this discrete optimization problem as a continuous optimization problem. Essentially what it does is it learns an underlying weight matrix here that is the same size as the sequence that we are trying to edit, a weight matrix of probability distributions. Let's see how it looks like midway through the process. At this first position, the initial sequence is a G. Ledidi doesn't really have strong preferences for what characters should be there. And so when we add these two together and we sample a character from it, we get the G again. At this next position, the initial sequence is an A. Ledidi really thinks it should be a T, and so we end up sampling a T, and that's an edit in our sequence. We can then proceed all the way down the sequence, sampling the original characters usually, or sometimes sampling the edited characters. Now that we have a discrete sequence, we can run that through a model, get our actual output, compare it to the desired output as before, and run that backwards through the model. When we encounter the edited sequence, we go straight through it and we update this underlying weight matrix. This ends up being quite simple. It's only a few lines of code to implement, and any off-the-shelf optimizer will work. What's a little bit more challenging is the loss function that we are optimizing. It is made up of 2 terms. The first term is kind of this traditional output loss. How closely are we matching the desired output? The second is this novel input loss term, which is quite literally the number of edits that are being proposed. And so by explicitly optimizing both of these together, Ledidi is saying that I want to use the minimal number of edits to an initial template sequence to achieve our desired objective. And then there's a mixture term because there are always 2. So sometimes people confuse, when they talk about model-based design, they confuse the machine learning model that is being used with the design method. They will say that they have a new design method that's really cool, when what they really mean is that they have trained a new machine learning model that's really cool and have used off-the-shelf design methods paired with it. Ledidi is a design method, which means that it can be paired with any model that's already public and on the internet, any model that you have in your lab, anything that you want to train in the future. You don't have to do anything special with your model in order to use it. And so in order to demonstrate that, we applied it broadly. We started off by applying it to a BPNet model that makes predictions for the binding of GATA2. We took regions where GATA2 was not really binding, the 50th percentile across the genome, shown in the black line there, and we wanted to edit these sequences to exhibit strong binding, 99.5th percentile signal, shown by the cyan line there. The filled in histogram there shows the distribution of sequences that are generated by Ledidi, showing that we have achieved our objective. But we can then apply this broadly across both different types of transcription factors and also different sorts of models. We can then look at something that's a bit more complex in terms of logic, chromatin accessibility, and see that Ledidi off the shelf with the default parameterization basically works on all four of these models to design accessible regulatory elements. We can then look at something even more complicated, transcription and transcription initiation, and see that Ledidi, again, with these four models, is able to achieve its desired goals. We can look at MPRA activity as predicted by the Malinois model. We can look at enhancer activity as predicted by the DeepStar model. Across all of these models, it just works at designing things that achieve the desired objective. One of Ledidi's strengths is that it is very fast. You can imagine that if you were doing design, one of the simplest methods you could use is just go to the Jasper database, take all 2,000 so motifs, and just try plugging them in at every position in your sequence and take the one that achieves the best objective. Doing one iteration of this takes over 1,000 seconds. If we limit this down to 14 motifs that we think are relevant for the task, this takes only around 40 seconds. If we look at a greedy substitution approach, we only make one edit at a time. This takes around 30 seconds. When we use Ledidi with various batch sizes here in order to test its robustness, it takes between 2 and 8 seconds to achieve. So this is around an order of magnitude faster. And this makes sense because with greedy substitution, you're trying to make all individual base pair changes one at a time, saying, does this help? Does this help? Does this help? Kind of like a petulant child. Whereas with Ledidi, at each iteration, you're asking the model, what edits should I make here? And then potentially making multiple edits in each round. Because Ledidi is so fast, it can be paired with multiple models simultaneously to design sequences that have really sophisticated characteristics. There is no real restraint on what sorts of models can be used. If you have a multitask model, you can use multiple tasks from it, like AlphaGenome or Informa. If you have multiple individual models that each predict a single thing, you can simultaneously use those. Maybe you have some models that predict profiles and some that predict counts. All you need to be able to do is define a desired output from each one of these models. And the way that it works is basically that you sample an edited sequence, you run it through each one of these models, you compare the predictions from that model to the desired predictions from that model, you run those backwards, you aggregate that into a single gradient update, and then you update the weight matrix just as before. So given this capability of being able to design elements using multiple models, the question is, which multiple models should we use? And so I'm at the Research Institute of Molecular Pathology in Vienna. I'm working closely with Alex Stark, who invented StarSeq. And so I was contractually obligated in order to use his DeepStar models. So we use DeepStar models in order to design cell type specific enhancers. We had these two cell lines. These were models from previous research projects. We did not have to train anything new for this research project. And we wanted to design regions that were specific to one of the two. And so because Ledidi involves editing an initial template sequence, the regions that we chose to start off with were important. The simplest strategy is to start off with regions that were enhancers in neither S2 or OSC cells. To start off with regions that were enhancers in neither cell line and try to turn them into enhancers in one cell line or potentially in both. And so when we take these non-enhancer regions and we turn them into regions that are S2 specific enhancers, we can see that they yield S2 specific StarSeq signal. This is an experiment that was performed by Franzi and Alex Stark's group. So this is the real experimental signal, where the x-axis is the signal in S2 cells, and the y-axis is the signal in OSCs. Other than a few outliers, we see that the OSC signal has been quite contained. When we take these same regions and we try to turn them into OSC-specific enhancers, we can see there's basically the same thing, except transposed. When we take the same regions and we try to turn them into enhancers in both cell lines, we can see that we're basically on the diagonal. Now, you may be looking at these results and saying, Jacob, that's not very impressive. You can see that many of them have quite weak activity, that the bottom left is where most of the activity is happening. I omitted a detail when I described this setup. There's actually a third axis here. The first axis is the initial regions that we chose. The second axis is the cell line that we want activity to be in. And the third axis is the strength of that activity. So we are intentionally trying to design a spectrum of enhancer activities. And when we group the regions by that label, we had 5 different categories here. We can see that not only is Ledidi able to qualitatively design cell type specific enhancers, but it is able to quantitatively define the strength of those enhancers. So here are the regions that were supposed to be S2 specific, exhibit a range of S2 activity, while limiting the activity in OSCs, which are in green here. There is a little bit of an issue here with the min gap loss. I'm not going to get into it, but you should ask me about it later. When we look at the same regions for the design to be OSC specific, we see kind of the same pattern, limiting the S2 specific activity while keeping the OSC active. And then when we try to design regions that are active in both, we see perhaps the cleanest pattern here of designing regions that have variable activity. So I mentioned that the initial regions are important. And so we didn't just consider regions that weren't enhancers in either cell line. We also considered regions that were initially S2 enhancers, initially OSC enhancers in both, and totally random DNA. We're still analyzing all of the results here. We just got the data. But the things that are the most interesting to me are these regions that started off as an enhancer in one cell line, and we edited to be an enhancer in a different cell line, because this involves 2 operations. The first is turning off the activity in the initial cell line, and the second is adding in the activity in the second one. And so here, I'm going to show you an example of an S2 region that was edited to be OSC. The predictions show that it transitioned correctly, and the experimental data also confirm this. So here, initially the model says that this region was an S2 enhancer because of the presence of these 2 gather motifs that are facing each other right here. Then, when we wanted to convert it into a weak OSC enhancer, we made 1 edit in the gather motif on the left to knock it out. We made 2 edits on the gather motif on the right to knock it out, and then 2 edits in the middle there in order to begin adding in a weak traffic jam motif, which is OSC specific. When we wanted to turn this region into a middle strength OSC enhancer, we can see the same thing on the flanks. But what's really interesting is in the middle, we are now only making 1 edit. It only took us 1 edit to go from no activity to a traffic jam motif that induced cell type specific OSC activity. It took 2 edits to then make that motif weak. So sometimes you may find some things that are quite surprising. Not everything is linear with respect to the activity here. Strong. More edits are necessary. I'm going to skip over this just because I think I'm kind of running out of time. And so in summary, Ledidi is a method that designs DNA by turning any machine learning model that's out there into a designer of DNA edits. Because Ledidi is so fast and flexible, you can use any model or set of models in order to design really sophisticated, complicated designs. But I want to take a little bit of a step back again. Our field has been designing state-of-the-art models that predict a wide variety of things, and people have been showing that these models have picked up on really sophisticated cis-regulatory logic. What's a little bit understudied so far is what we do with these models once they have been trained. Is it enough to just train the model and show that the predictive performance on some reference data sets are high? Something that's really interesting to me is developing methods that make use of these models in order to do something cool. Laura is about to give a talk next about another such method that does this, and I really encourage you all to stay and listen to it. But if you agree with me about the need for developing such models, I'm about to start a position at UMass Chan Medical School. I know that now isn't exactly the best time to be advertising a position in the United States. But one of the things that I can offer the Europeans in the audience is that we do have air conditioning. So I'd like to thank everyone who helped me along this journey for these many years, and also thank you for your time. Thank you.
So we do have time for questions.
Hi, thanks for the great talk. I was curious about ways to avoid particular sequences that might present adversarial examples to the guiding models. Do you have any thoughts on that?
Yeah, so one of the slides I didn't have a chance to show is that when you use multiple models, typically you think that each model should represent something different. If you want to do more robust design, kind of like what you're talking about, one of the things you can do is use multiple models that all make predictions for the same thing, so that the edits that you make have to be agreed upon by all of the models that are being used. So you're bringing up a great concern with design. You don't want to push the models out of distribution, and there is no silver bullet that completely solves it, but this sort of robust design actually can help quite a bit.
All right. Great and fun work, Jacob. So for your output loss, are you able to I mean, expressions, I think, very trivial, but different chip-seq tracks, attack-seq tracks, maybe things that are fractional, like methylation, all of them, are you able to handle?
Yeah. So
yeah,
I mean,
whatever is being predicted by the, the model, underlying model, I guess.
it really depends on whether or not the model can make predictions for it, and you can define a loss function. For most of these, I use mean squared error loss, but you don't have to use that. You can use any loss that's differentiable. You can be really creative in what you do.
Thanks.
Hi, Jacob. Great talk. Can you help understand? So chip-seq, I think it's easier to understand, but in chromatin accessibility, for example, it seems like maximum accessibility can be achieved in multiple different directions. You are choosing greedily one path.
Yeah.
What is the interpretation of this specific path? Is it arbitrary, or what are you optimizing? Are you missing some other interesting things?
That is a great point. There are methods out there that do design that focus on diversity, being able to generate a wide variety. Ledidi is not one of those. That when you run Ledidi once, you get basically one solution. Because it is fast, you can potentially run it many times and each time get a different solution, but you should not interpret the design as the only possible way of achieving that goal. But there are quite simple ways of using Ledidi in order to, by running it multiple times, each time getting a different answer.
Thank you, Jacob. Really great talk. I would like to follow up on that and your answer. Do you have any ideas about actually the amount of possibilities given a certain sequence, right?
Yes.
Yeah. Did you look into that, basically?
This is something that I have looked into briefly in a single Jupyter notebook. And so the idea of being able to represent a sequence based on how editable it is in a certain direction is a really cool direction that is outside the scope of this work, but absolutely something that I think is worth looking into. Because so much of the genome is almost active. It's only a few nucleotides away from going in a completely different direction than it ends up going. And I think this is something that's really interesting to think about why it is that case.
And then when you have your edited sequence, have you looked at how much does it correspond now to a known sequence that you've already trained on, basically? Are you reproducing things that it's been trained on, or are you always very far away, or how far away is it? I don't know. Do you see what I mean?
Yeah, this is not something that I've explicitly looked at because we are trying to minimally edit a region. And so, of course, it's probably going to be quite similar to the region you started off with. But in my view, I don't really care so much, even if it did totally recapitulate a sequence that it saw before. If it works, that's the goal, right?
So thank you, Jake, for being with us.